On behalf of the CytoData Society, it is my pleasure to welcome you all in this CytoData 2020 event. So in my presentation I will briefly describe the CytoData Society and give some recent advances in image-based profiling. The mission of CytoData Society is to build and maintain an active community around image-based profiling, and what we are now doing these 2 days is one part of our tasks. So we organize an annual symposium, and together typically with hackathons, as well as organize interconnection between the members in academia and industry, and also collaborations between the members in academia and industry. Something what we will launch in coming months is a webinar series to disseminate this information and knowledge available in the image-based profiling. A brief history of the society. So the first event called Cytomining Hackathon was organized at the Broad Institute in 2016 by Carpenter Lab, where approximately 20 different labs were invited to participate and discuss strategies that are used in these different labs. Then next year we met at London for a first symposium and friendly competitive hackathon. And then early 2018 the CytoData Society was founded to organize these events as well as the community around the image-based profiling. So after that there's been two symposia afterwards, 2018 again in Broad Institute, and last year in Heidelberg at the German Cancer Research Center. And this year unfortunately we cannot be at the Salt Lake City physically, but then I'm very happy that Recursion took this challenge to organize a virtual event for us. What will then happen next? We will start the monthly webinar as mentioned, and then also organize the application period for the CytoData 2021 event. And these events have been organized every second year in US and every second year in Europe, so then the next time it will be in Europe. It's still open naturally whether it will be a virtual event or physical or some sort of a combination of these. Some photos from previous events. Top left is a photo from the first event at Broad Institute, where we were about 30 of us participating the event. Then the lower left photo is from the hackathon last year in Heidelberg, when already approximately 40 researchers participate the hackathon, and altogether 150 to the whole event. And I think this year we have a record number of participants, over 200 for this virtual CytoData event. Unfortunately, we cannot meet physically and go for lunch or beer this year, but then hopefully already next year we can organize these events. But it's been a very warm and welcoming field and community built around the image-based profiling, so I welcome you everyone to also join these physical events in the future. And also one company was formed during this or due to this collaboration started from the CytoData events. So the current board of the society looks like this. We have a couple of open positions, as well as some of the members are currently responsible for multiple positions in the board. So if you are interested in joining this great group of people and work at the core of the society, then check the website society.cytodata.org and apply for these open positions, and we will organize voting for the whole community to select the board for the next year. One paper that resulted from the first meeting at the Broad Institute was this data analysis strategies for image-based cell profiling paper, where we put together these different image and data analysis strategies that labs working in image-based profiling were using, and gave an examples for various different tasks that can be used for profiling the data, and also identified some challenges that the field had. One was the lack of standard methods. Basically all groups were working on their own set of methods and libraries, so that there are some common software that many libraries were using. Also, there's a lack of standard assays, and importantly, lack of large, open, and well-defined data sets. Also for organizing benchmarks. And due to the rise of deep learning, there's also a lack of general models that could be applied for different kinds of imaging datasets. By the time of publishing the paper, there was only a handful, maybe 10 papers in this field where deep learning was used. But then, as many fields working with images, there have been a deep learning revolution during the last few years. Especially, there's been general models for segmentation and cell detection. Also for various methods using deep learning for learning the representation of the data, and then extracting these representations for profiling applications. And then also deep learning has been used for this typical supervised approach for classification. But still, these many challenges exist, especially on the batch effects or plate effects or normalization of the data. There's been one competition organized for that as well. Then, especially when there's a small amount of annotated data available, very typical problem with deep learning is the overfitting. The model learns very well the data available, but then when you are trying to apply it even in your own experiment for other experimental batch, then it doesn't anymore work for that. And then some very practical problems, for instance, extracting these embeddings of 2000 features for each channel can easily result, for example, as using five channel dataset in this feature vectors containing 10,000 features. So this might result in feature matrices that are actually larger than the raw imaging data. Other fields of science, especially in the computer vision, they had ImageNet datasets already more than 10 years, and the competition regarding that dataset, even though that the competition is not anymore organized in its original fashion, but it moved forward the computer vision field very rapidly. And I'm very happy to see that in our field also there's many options for open datasets nowadays. So broad bioimage benchmark collection have been already available for some time, but new datasets are continuously shared in BBBC. Then Image Data Resource, which was opened a couple of years ago, which is not only for image-based profiling field, but then all sorts of imaging data, but then also including meta information of the data. So it can be used for these image-based profiling method developments. And then Recursion opened last year some of their data in this rxrx.ai platform, and there's already now more than 1 terabyte of data available there. Then about the standard assays, so during the last few years, cell painting assay have emerged to be a sort of a standard assay in the field, utilized by many research labs as well as industry. And I'm very excited to hear about this JumpCP consortia and the massive open dataset that they are creating there. There will be also a presentation about that in this symposium. And also few competitions have been organized in the image-based profiling field. In 2018 there was a Kaggle Data Science Bowl for nucleus segmentation from various imaging modalities. Then Human Protein Atlas image classification competition, where localization of proteins were classified. And then Recursion organized also image classification competition focusing on dealing with patch effects. And these have been very essential of moving the method development forward in these rapid sprints. Some results from the first DSP 2018 nuclei segmentation challenge. So we published this general model for segmenting nuclei called Nucleizer that will be presented in this symposium by Reka Hollandi. And then another example of these general models for segmentation from Janilia called CellPose, which is taking a challenge of segmenting whole cell, so for instance the cytoplasm of cell, and sharing this general model as well as website for testing this method. And explorative methods and various way of learning the data have been the core of the image-based profiling field from its origin. This one visualization of UMAP visualizations of drug mechanism of action clustering, which can be also used for quality control for finding these patch or plate effects. And some examples of methods published recently for these various different ways to learn the data. So Caicedo et al. Presented this weakly supervised approach using meta information available for as labels for the training task. Then I think the 2 other methods will be presented also in this symposium. Lu et al. Develop a self-supervised approach for learning the representation of the data, and Janssen et al. Develop a fully unsupervised method built from clustering and then convolutional neural networks for learning the mode of action of the data. And some applications, so image-based profiling is applied in drug discovery, genetic screening, also for finding treatments for COVID-19, and in other fields, for instance the pathology, these similar methods can be used for profiling that kind of a data sets, and then for toxicology, for instance. So there's a wide range of different applications where these image-based profiling methods can be applied. So some conclusions. So the community is working to solve these challenges identified in 2017, and we've recently seen many of these new general models published, and also open datasets and creation of a new reference datasets. Then these competitions have been very important for the field to have these fast bursts of method development. Hopefully we see those in the future as well. But then there are new challenges also coming in the field, especially in the form of 3D assays and these highly multiplexed datasets, once the technologies move forward to be able to produce data out of from a huge number of samples. So I hope you all enjoy these 2 days in the CytoData 2020 event, and thanks to Recursion again for taking up the challenge to organize this virtual event.